Hello everybody, and welcome to an exciting edition of Ed Puzzle Notes. Today we're going to go over what's called the scientific method. Scientific methodology. That is a systematic, orderly method of gathering data to solve a problem or to answer a question. Now I have the problem that I really am not very good at Fortnite. In my whole career, I have got one win. And in that win, I was playing squads with my nephew's friends. I got 0 kills and basically just hid the whole time. So I don't know if that win really should count. But I now have a hypothesis that I want to test. My hypothesis is, you can see the guys right here jumping out of the bus. Usually they go straight down to get to the ground as fast as they can. But my hypothesis is, if I can fly as far away from the bus as I can and get as far away from everybody else as I can get a lot of guns and I won't get killed. So my hypothesis is, fly as far away from everybody else as you can, loot a bunch of houses, get a bunch of good weapons, and I'll have a chance. Well, that is what I actually do when I play. Every single time I play, I fly as far away as I can. That is what I'm testing. That's my hypothesis. I think that the farther away you fly, the better. So what I'm testing, that's called my independent variable. I am testing what happens when you fly as far away from the bus as possible. Now, what am I going to be looking for? So say I play 10 times. And every single time I fly as far away from the bus as I can. What am I going to be looking for? I'm going to look for how well I place and how long I stay alive. So those things that I'm measuring, like what place I get, that's called the dependent variable. So the scientific method is really just a way to test your hypothesis. Is it true? Is it true that flying as far away from the bus is actually better than just diving straight down? And in this case, how far away from the bus you fly, that's my independent variable. And how well you do, how many wins you get, that would be called the dependent variable. Okay, so here are some steps of the scientific method. Once again, that's just an orderly method of gathering data to solve a problem or to answer a question. On the previous slide, my problem or question was, how can I get better at Fortnite? So that was me defining the problem. Now, we have some problems down here, or some questions. Here's a question. Does wearing track shoes actually make you faster? That's a question. That's a problem. I'm assuming that they do because, number 2, I've made careful observations. And every time I see a track meet, whether it's at Tulare Western or in the Olympics or whatever, I always see track stars wearing track shoes. Now to test that, whether track shoes actually work, I can have this sprinter right here. I can have her run a 100-meter dash in track shoes and then have her run it again in tennis shoes. That would be a way to test it. Well, in this case of the female runner, the data I would collect is how fast does she run? How long did it take for her to run 100 meters? In this instance, since there's a number, since I can measure it, since I can measure how fast she runs, that's what's called quantitative data. Those are observations or data that can be expressed as numbers. So, how fast she runs, or how many wins I get in Fortnite, that would be quantitative data. Here is another little example right here. Now, what would happen if you gave your flowers orange juice instead of water? Would the flowers smell better? That would be a hypothesis. Giving flowers orange juice instead of water would make them smell better. Well, since you can't really give a number to smell, you can't say like, oh, the flower smells like a 3. You can just say it smells good, bad, smells okay. that wouldn't be quantitative data. That would now be qualitative data because that is a description of what you are observing. So for these flowers, how well they smell, that would be qualitative data. All right, a super important term is the hypothesis. That is a proposed explanation of what you are observing that can be tested. It is usually written in an if-then statement. So it would be something like this. If I jump far away from the bus in Fortnite, then I will live longer. If I wear track shoes, then I will run faster. If I feed my flowers orange juice, then they will smell better. All right, so step 4. Now that I've defined the problem, I figured out the problem I want to solve, or the question I want to answer. I've made observations, and I've made my hypothesis. Remember, my hypothesis is my guess as to why something occurs. Now what you do is you're going to test your hypothesis by performing an experiment. Now, in a controlled experiment, we have things called variables. And there are 2 types. The first one is called the independent, or it's also called the manipulated variable. This is the variable which is being deliberately changed by the experimenter. This is the thing that is actually being tested. So in Fortnite, the independent or manipulated variable is how far away you fly from the bus. That is what I'm actually testing. For the sprinter, I was testing whether or not her track shoes made her faster. So it was the track shoes that was the independent or manipulated variable. With the flowers, it was the orange juice. I was testing whether the orange juice would make the flowers smell better. Now the second variable is called the dependent, or it's also called the responding variable. This is the variable that you're actually measuring. This is the one that you're seeing what happens when you do the independent/manipulated variable. What happens when you wear track shoes? What happens when you fly far away from the bus? So, the dependent or responding variable is the conditions which is caused by the independent variable. So, for example, in Fortnite, how long do you live, or how many wins do you get? That would be your dependent or responding variable that you're measuring. For the track star that's wearing the track shoes, how fast she runs the 100-meter dash, that would be the dependent or responding variable. For the flowers that you give orange juice to, how well they smell, that would be the dependent/responding variable. Now, in a controlled experiment, we always have to have what's called a control group. This is the part of the experiment, or the group, which does not get the independent variable. For example, if we want to figure out if those track shoes made that female sprinter faster, we also had to have her run with no track shoes. No track shoes. We got to make her run in tennis shoes. Because if we don't, how will we ever know if the track shoes made her faster than if she's not wearing track shoes? So when she doesn't wear track shoes, that is the control group. Same thing with the flowers. We gave some flowers orange juice, and we gave the other flowers water. The flowers that just get water, that's the control group. Why do we need it? Well, how will we know if flowers that we give orange juice to smell better unless we can compare them with the flowers that just get water? So we always need a control group in order to compare to see if the independent variable actually worked. Now, some people get these confused, but there's also what's called controlled variables. It's a little bit different. It's different than the control group. Controlled variables is just within your 2 groups. So, for example, the flowers. Some flowers we gave orange juice to, the other flowers got water. And we wanted to see if the orange juice flowers would smell better than the flowers that just got water. Well, the only thing you're going to change is whether they get orange juice or not. And that's your independent or manipulated variable. Everything else has to stay exactly the same. So, One group of flowers gets orange juice, the other gets water. But they have to be the same type of flowers. You can't have roses in one group and daisies in the other. They both have to get the same exact amount of sunlight. They both have to be in the same type of soil. They have to get the same type of fertilizer, etc. The only thing you can change is your independent variable, and in that case, it was the orange juice. Because if you change another variable other than just the orange juice versus the water, you won't know whether the flowers smell better because of the orange juice, or do they smell better because roses just smell better than daisies. So that's why you can only change one independent or manipulated variable at a time. You cannot change 2 or 3 because then you will not know which one made the difference. And the final 2 steps, make your observations and collect your data. So basically, hey, what happened? How well did the flowers smell? Do they smell good? Do they smell bad? How many times did I win in Fortnite when I flew really far away from the bus versus when I just flew straight down? How fast did I run in the track shoes versus the tennis shoes? That's me collecting my data. Then, when I'm all done, you make your conclusion. Do track shoes actually work? Does orange juice make flowers smell better? Does flying really far away from the bus in Fortnite help you win more often? Okay, so a little trick to help you find which ones are the independent or dependent variable or the manipulated or responding variable. Well, this week or last week at school, you all took pictures to get your ID card. Well, everybody knows that I comes before D in ID. Also, if you have a teacher that's a male, you probably call him Mr., hopefully. Well, if you write a proper hypothesis, the independent variable or the manipulated variable, they're both the same thing, those always come first. Just like the I always comes before D, and the M always comes before R in Mr., the independent and manipulated variable always come first in your hypothesis. Now, the D comes second in ID, and the R comes second in Mr. Well, the dependent or the responding variable, they both mean the same thing. These always come second in the back part of the hypothesis. For example, here we have a hypothesis. If students eat mints before exams, then they will score higher. Well, the independent/manipulated variable, that's always what you are testing to see if it works. In this case, we're testing to see whether eating mints will make the students do better. So mints is the independent/manipulated variable. Now, what are we actually measuring? What's the dependent or responding variable? We know it's going to be over here. Well, we're going to see how high they score. That's what we're going to measure. So mints is what we're testing. That's the independent or manipulated variable. How high the students score, that's the dependent or responding variable. Now, what are some controlled variables between these two groups? Because we're going to have two groups of students. Some students are going to get mints to eat before they take the test, and some students are not. Well, the most obvious controlled variable that you're going to have to do is the students that get mints and the students that don't get mints, they got to be pretty equal when it comes to how smart they are and how much they study. So for example, I cannot give mints to my honors class and then have a control group of my regular bios that don't get mints, because does that really prove anything? Usually my honors score a little bit better than my regular bios. So that would be a controlled variable. Another thing that would have to be controlled is how hard is the test? I can't give a really, really easy test to the people that eat mints and a super hard test to the people that don't get mints. That would not prove anything. That doesn't prove mints work. That just proved that the test was way easier for the people that ate mints. All right, let's see if we can do this one. If you read before bed, so we're doing this right here. If you read before bed, then you will fall asleep faster. All right, first let's try to find the independent or manipulated variable. We know it's in the first part of the sentence. What is it? Well, hopefully you said reading. That's what we are testing. What happens if you read before you go to bed? Now, the dependent or responding variable is always in the second part of the sentence. Let's see if you can tell me what that is. That's right. Hopefully you said it's how fast do you fall asleep? That is what we are going to measure. We're going to see how long it takes you to fall asleep. So reading, that's what we're testing. That's the independent / manipulated variable. How fast you fall asleep, that's the dependent / responding variable. Now, obviously you're going to need a control group. The control group is going to be people that do not read before they go to bed. Because we won't know if you go to sleep faster by reading unless we can compare it with people that don't read before they go to bed. Now, some controlled variables. The people that don't read before they go to bed, obviously they can't drink a lot of coffee. More coffee than the people who do read. Because maybe those people can't sleep just because of the coffee and not because they didn't read before they went to bed. All right, last one. If you feed your dog Gatorade, then it will have more energy. All right, first thing, tell me where is the independent or manipulated variable in the first part of that sentence? What are we testing? You are right. We are testing Gatorade. We're going to test Gatorade. Now, what is the dependent or responding variable that's going to be in the second part of that hypothesis? Let's see if you can tell me. Once again, you're right. How much energy the dog has. So we're going to test, we're going to give the dog Gatorade. Don't do this at home, by the way. I just made this up. And then we're going to measure how much energy the dog has. Now, obviously, controlled variables, you got to use the same type of dogs. You cannot give the Gatorade to a very hyper type of dog like a Chihuahua and then maybe a Basset Hound that likes to sleep all day, give him water and then say, yay, Gatorade worked. Well, no. You got to have the same type of dogs. The dog's got to get the same amount of sleep. They got to eat the same type of dog food, etc. All right, now here's a very famous controlled experiment conducted by a man named Francesco Redi back in the 1600s. Now, way back then, it was way before refrigerators, and they would always notice that maggots, the little worm-like organisms, would always appear on food. And most people thought that they just came by spontaneous generation. Basically, the maggots just came out of nowhere. They just generated from space. But Redi made some observations. His observations was he noticed that flies would land on the meat. And that later on, after the flies landed on it, he would see that maggots would appear on the meat. Now, what do you think Redi's hypothesis was? He observed flies landing on the meat, and then later he saw that maggots would appear on the meat. What do you think his hypothesis was? Remember, try to put it in an if-then statement. Now, hopefully you wrote something kind of like this. Redi believed his hypothesis was, if flies land on meat, then maggots will appear. Now, what Redi did to test this was pretty straightforward, pretty simple. He got two groups. He had a control group of meat that was in jars that were uncovered, so the flies could get in there. Then he had the experimental group, the same type of meat, but this time he covered it with a cloth covering where air could still get in, but the flies, they couldn't get in there to touch the meat. He then waited several days to pass, and he made his observations. He saw that the jars that were uncovered that the flies could get in had maggots all over the meat, where the meat that was covered and the flies couldn't get to them, no maggots appeared on that meat. So Redi's hypothesis was confirmed, and his conclusion down here was maggots form only when flies can come in contact with the meat. That was his conclusion. Now let's see. Can you guys come up with any controlled variables? What had to remain the same between the uncovered jars and the covered jars? See if you can think of a couple of those. All right, so real quick, some controlled variables. Obviously, in both groups, you have to have the same type of meat. You can't have pork in the uncovered jars and beef in the covered jars. You have to have the same type of meat. Both groups of jars have to be in the same temperature. You can't put the covered jars out in the snow and then the uncovered jars inside your house. It's got to be the same temperature. Also, obviously, you need flies. You got to have flies in both groups, even though the flies don't get into the meat on this one. Everything has to stay the same. The only thing that Redi changed was the covering. In one of the groups, he covered the jars, and the other one, he did not. Everything was kept exactly the same, and that's what's called controlled variables. Now, what was Redi's manipulated variable? Well, we know that the manipulated or independent variable always comes in the first part of the hypothesis, and in this case, what he was testing was whether or not flies land on the meat. What would happen if flies land on the meat? That is his manipulated variable. His responding variable, or dependent variable, always comes in the second part of the hypothesis, and what he's measuring is, do maggots appear on the meat? So that was his responding variable. All right, I apologize. This is a lot longer than I thought it was going to be, but we're almost done. All right, scientific theory. Now, in everyday language, a theory is usually like your guess. But no, that's what a hypothesis is. A hypothesis is your guess. A theory is not a guess or a hypothesis. A theory is a very strong statement that is backed by tons and tons and tons of research or experiments. And theories state how or why natural events occur. Now, the most famous one in biology is Darwin's theory of evolution. So even though some people, don't believe in evolution, that's fine. But evolution is backed by tons and tons of research. So it is no longer a hypothesis. It is a very strong statement, it is a theory. Now, very, very similar to a theory is what's called a scientific law. That is also a very, very strong statement that is pretty much accepted to be true. The only difference is scientific laws do not attempt to explain why it occurs. And a famous one of that would be Newton's law of gravity. He knew that the moon and the earth were attracted to each other because of gravity. But Newton did not know why, and he did not try to explain why. That's why Newton's was just a law. Even though it was a very strong statement, it also was not a hypothesis, which is a guess. All right, since this is so long and I wanted it to be way, way shorter, I'm not going to talk about these. Just get these in your notes. Honestly, we're not going to really need to know them that much for this unit. We'll talk about them again in later units, so just write them down. Push pause if you have to, because I'm not going to say much. And same thing with this slide. These terms are going to become really important when we get to ecology in a couple weeks. Write them down in your notes, but I will explain them more in detail later. All right, thank you for listening. Sorry it was so long, but I'll talk to you next time.